Hi, everyone. And today I'm doing the first introduction of our spring semester, and I have the pleasure of introducing Dr. Brad Sutton. He received his undergraduate degree in general engineering from the University of Illinois. So yes, he knows this place very well. He then received his MS in electrical engineering and biomedical engineering and his PhD in biomedical engineering from the University of Michigan. He returned to Illinois after his PhD, and he served 3 years as a research scientist for the Biomedical Imaging Center at Beckman. He joined the Department of Bioengineering here at UIUC in 2006, where he's currently a professor and the technical director of the Biomedical Imaging Center at Beckman. And he's an affiliate of the Neuroscience program in the Department of Electrical and Computer Engineering. Dr. Sutton's research is in developing MRI techniques. MRI techniques have been applied to examine the changes in the brain that accompany aging and examine the dependence on the brain microvasculature of age-related declines in brain structure and function. So without further ado, please give a warm welcome to Dr. Sutton, and thank you so much for giving the seminar today.
Great, thank you very much. So I hope you can see the slides. Yeah. Yep. All right. So thanks for this opportunity. Glad to be the person who can lead us off on a new semester here, and even in a virtual format still, but hopefully this is the last one of those for a while. So I'm going to talk about some work that we started doing recently, leveraging some technology built in Beckman Institute here by Xipei Liang a couple of years ago too, and we've been able to leverage it for a variety of other purposes, and very glad to see that he's on here too. So we're going to talk a little bit about fast fMRI, some techniques we've been doing and what we've been trying to use them for, and certainly glad to answer any questions that people have as we go too. So please just go ahead and interrupt me and ask. So I'm going to give you, of course, the little spiel, the sales pitch on MR technology in Beckman Institute 2. We've added a lot of tools. I want to just quickly point those out. Some of you have heard this from me before, but some people it's new. Then I'm going to tell you a little bit about how functional MRI works so you can understand what it is we're trying to image. Then we'll say how we can go faster, and then we'll figure out why we would want to do that. And I will tell you up front, this is new work for me, trying to figure out still why. And I'll show you then how we are going to apply this in another scenario at the end too. All right, so a little bit update on MRI technology on campus too. And so really exciting times here on campus in terms of imaging technology, and really excited to see what the future will hold too. We have so many things going on with a new cancer center coming and all sorts of stuff down the road. What an exciting time to be here. And I encourage you all to take as much advantage of these things as you possibly can. We have two state-of-the-art 3T MRI systems in BIC, but over the pandemic here, we've added a 9.4T 30 centimeter animal MRI system shown here, and a 7T in partnership with Carl. And these are really unique technologies that will allow us to unlock new neuroscience problems and answers, and be able to see things like we haven't been able to see them before. And we add to our existing technology. We have a PET spec CT system in BIC also, and a fluorescence system too. So it's a really great complement of technologies to go see exactly what you're interested in for your neuroscience question. And there's lots of other MRI in town, and it's growing too. Carl just completely revamped the radiology department. And so they bought 3 new scanners, and in addition to, they had another 3T Prisma and then the 7T, all in the same hallway now. It's pretty exciting to see that set up there too. So this is our animal scanner, and you can do great work. We've been working with Martha's group on several things too, and doing lymphatic flow and looking to see how contrast agent moves around in the brain. A lot of other groups interested in very interesting things too. We have fMRI of birds coming up soon, and looking even at non-biological samples, looking at batteries and the like there too. This is an example of an octopus arm, and looking at the diffusion signal associated with that. It's really a bunch of muscles that are arranged in different directions to be able to let the octopus move however it wants to. And this is exciting work going on there too. This is our 7T system, and it's sited at Carl, and producing just beautiful images. This is the actual screenshot I took with Aaron Anderson's feet sticking out of the scanner. It's pretty amazing the types of images you get. These are almost like the cartoons in a textbook, that they show the structure so well and with such high contrast. And so we own this 3.5 days a week, and Carl owns it 3.5 days a week. It's great that it's sited in the clinic because we can run clinical studies and get patients in the scanner too. So it's one of the first 10 FDA approved 7T systems in the US. And so we can actually run patients in there for their clinical study, and we can tack on research studies at the end. We have a population study going right now, a collaborative study between the two universities. If you want to see the MRI scanner from the inside, please visit this link and you can get scanned. Okay, so sign up, and we're trying to scan a bunch of people in the first couple of years here to show how good the technology is and how much more you can see at this high field 7T than you can at a 3T scanner. And it produces wonderful images. So high resolution structural images. This is looking at iron composition in the tissues, and this is really where the machine shines. And then diffusion imaging too, looking at all the different connections. And then I just wanted to point out too, we have pipelines in place where we're we're trying to leverage. Yeah, so Aaron put the link to the study in the chat too. Thanks for doing that. We have pipelines that are being developed by neuroscience graduate student Paul Camacho that are being run on everything to produce all sorts of outputs, connectivity maps, and lots of different atlases. And so functional connectivity, structural connectivity, and really leveraging the system, but also applying these techniques to all the data coming out of BIC now at 3T2, and really applying this high throughput computation to be able to get all sorts of results so that you know immediately after you scan how well, what the quality of the data was for the purposes that we want. We want to know what parts of the brain are talking to what. And I'm going to talk a little bit about functional connectivity in this talk. So this is truly a partnership between Carl and Beckman, and this is the Carl Illinois Advanced Imaging Center. So it's being led by people on both sides, and a true partnership there. And then finally, my last bit of my sales pitch here. Okay, so apologize for the sales pitch here, but please do take a look at our MRI museum on the first floor of Beckman Institute too. Really, we're building all these things on top of such a great and rich history in the development of MRI. And so of course we have Paul Lauterbur here, who won the Nobel Prize in 2003. But there was a lot of other people who came through Illinois that were responsible for the invention and then the commercialization of MRI technologies. And really Illinois is the key place that all these things came through to get us to where we are today. And we lay out a lot of that in the museum. So please take a look. We have Paul's original first 2 human MRI systems from the 70s. We have his data, the very first, we found his data notebook, the very first data showing the MRI worked, and we have that all on display. So please do take a look at that too. All right, so thank you for humoring me there. Please take advantage of these opportunities and please take a look. So now I'm going to tell you a little bit about this particular work that I'm talking about today. And first, to make sure that you understand what we're doing, I know it's a pretty broad group. I'm going to explain how functional MRI works, and then we'll go into what the specific technology we're developing is here. So there's quite a few ways you can image brain function with MRI. When we're just taking a picture of a brain, you really can't see the function. So we need to have some type of contrast that shows us which parts of the brain are active during the performance of a particular cognitive task and which ones are not. And so the most common technique we look at is called BOLD, B-O-L-D, and it's blood oxygenation level dependent. Okay, so it depends on the blood oxygenation level. And we'll talk about that in a minute. And that's our most common one, but there are other ways that people have tried to image brain function. And so we can look at changes in blood flow. And so we do that here too. And when a part of the brain is activated, when it's performing a cognitive task, the brain vasculature responds by sending more blood to that area. Okay. And so if you image changes in blood flow or blood volume, you can actually see the function show up. And we're going to see more of that as we go forward with 7T MRI, because imaging blood volume changes or blood flow changes is a key way we're going to get layer specific functional imaging to come out of 7T. You can also look at neuron size changes. There's been stuff done with diffusion to look to see how a neuron swells when it fires. And there's been some interesting data from MRI that shows that we can see that. Other metabolic changes. So if people went to, we have a 7T seminar series on Monday, Xipei Liang talked about some of the metabolic imaging that's happening here too. And you can see some turnover of metabolic components when activity happens or when pathology occurs in tissue. And that's a key area to be able to get really quantitative measures of function. And then there's other things that are a little bit farther out there that we're not sure whether they'll work or not, such as magnetic fields produced by active neurons. And there's work going on here with Sepeda Sedegiani, looking at some of these things too with a new take that hopefully will give some higher sensitivity to some of these things. So the main, though, way in which brain function is imaged with MRI is bold. And so blood oxygenation level dependent. And what happens is you have this blood in your vessels, in the capillaries in your brain. And that blood has hemoglobin in it, and that hemoglobin has iron in it. And so we have this large magnetic field, and we have iron in the blood, red blood cells. And when those red blood cells are deoxygenated, okay, which I'm showing some red blood cells here going through a capillary, the iron in those deoxygenated red blood cells can interact with the magnetic field, and it disrupts the magnetic field, okay? And what happens then is, if you're really close to this blood vessel, you experience a different magnetic field than when you're really far away. And it turns out in MRI, we're looking at the protons in the water and other chemicals, but mainly the protons in the water, and they spin at a frequency proportional to the magnetic field that they experience. And so if they're close, they're going to spin at a different frequency than if they're far away. And what's going to happen is they're going to get out of alignment and cancel. Okay, we're going to get a reduced signal. But when that piece of tissue is active, your brain vasculature responds by flooding that tissue with extra blood. So things dilate, you get more blood. And what happens then is that the oxygenation level goes up in that tissue. And so we see here, I've got fewer of these deoxygenated components, and the magnetic field becomes more uniform then, because that oxygen shields the iron from the magnetic field. So what's going to happen is that if you're close to this or you're farther away, you're going to actually experience more of the same magnetic field, and you're going to spin at the same frequency, and you're not going to get out of alignment. So I have a couple of really bad simulations of this just to kind of show. I have a red proton and a black proton in my tissue, and they are spinning at different frequencies. Excuse me. I'll try not to cough into the microphone next time. And if we wait a certain amount of time, they get out of alignment with each other and they cancel, and the signal goes away. So if we excite them, we let them spin relative to the magnetic field they're experiencing, and we wait a while before we sample, they're going to give us different signal. So it turns out when they're active, the magnetic field is more uniform, and we excite them here and we let them decay away, and we image sometime later at our echo time, TE, and we're going to have a high signal. But when they're at rest, they have deoxygenated hemoglobin, they get out of alignment really quick, we excite them and they decay really quickly. And when we image at this TE, they've lost their signal. So if we give someone a task to do, and we have them not do the task for a while, then we have them do the task for a while, and we keep taking images all those times, we're going to see a difference in the signal across those two. And we're going to be able to localize which parts of the brain have this oxygenation effect, which parts of the brain are active during this task, okay? It's a very complicated relationship between the neurons being active and us getting this change in our image. And I want to point that out because there's lots of things that can happen. So there's metabolic changes, there's blood flow and volume and oxygenation changes. Those change the magnetic field uniformity. And then those affect the T2 star changes in the tissue, which eventually affects the signal that we get. But it's a very complicated pathway. And you can imagine that if people have different pathology, or as you get older, different parts of this system kind of work differently. So how much can you dilate the blood volume of your blood vessels? And that may depend on cardiovascular function and the like too. So there's some interesting things that can happen along here between neuronal activity and then the measure I can make with MRI to see brain function. And I encourage you to look at this primer if you're interested in more information about this. It's a really good primer, very easy to read. And you can Google that primer and it will show up. So, all right, so let's give somebody a task. Let's have them do something, and let's see what happens to that signal in the brain, okay? It's gonna go up, but it doesn't go up immediately. So when we give somebody a task, there might be an initial decrease, okay? And that might be because the brain is active and it's extracting some more oxygen before the vasculature system can overreact and respond, and then the signal will go up. And then the task is over, the vasculature then takes some time to return back to normal, okay? And so this is kind of the waveform we get, and these are seconds. So I give you something really fast. Let's say I'm trying to see whether you recognize a face or not, and I show you a face. I flash it onto the screen. It actually is a very slow process for me to see whether your brain reacted to that or not, okay? So it can take 20 seconds for the whole response to go. So it's not a very quick mechanism, and that's really important for what I'm going to talk to you about today, and what the value of what I'm going to talk to you about today is, because we're going to try to go really fast, but we cannot beat this signal. The signal is very slow, okay? So that's why when we get to it, I'm not going to show you really fast brain function because I can't change the physiology of the brain. I can only change the way I'm measuring it and what that means, okay? All right, and certainly if people have questions, I encourage you to ask. So I'm gonna show you an example of an fMRI task, okay? And this is going to be just to identify visual or motor areas. We may instruct the subject, watch this fixation cross. Okay, so this is time going in this direction. Watch a fixation cross for a while, maybe 20 seconds or so. We're gonna turn on a flashing checkerboard. Okay, and we're going to stimulate their visual cortex. They're supposed to keep looking at the plus sign, and maybe they're supposed to tap their fingers while they're looking at the plus sign. I'm just going to show you visual results here. So just watching the plus sign, and then it goes to fixation cross, and then it comes back again. And like I said, maybe 20 seconds off, 20 seconds flashing, 20 seconds off, 20 seconds flashing, and we repeat that a couple of times. And then what we can do is we can look at the brain, and we can look for pixels in the brain at every single voxel of the brain and look to see, does its signal change with what I would expect from this task? Does it stay low and then go high and then low and then high? And I can make statistical maps based on that, and I can threshold those statistical maps, and I get a functional activation map. So this is the visual cortex, and that's from a task very similar to what I just showed you, 20 seconds of rest, 20 seconds of flashing checkerboard. Okay, so we can make a map of where that occurs in the brain. And what we're doing is we're acquiring images, every 1 or 2 seconds, we acquire a whole image of the brain with this right weighting for this functional task. And then we look to see in every pixel what's happening relative to the task that we just provided. Okay, the problem with fMRI in this mode is that you have to carefully design the task for the specific region of the brain, the specific cognitive function that you want. And this can become very hard because some things are very complex and have multiple cognitive functions embedded, and you can't do one without the other. It can also take a really long time. If it takes me 3 or 4 minutes, I have to turn things off and on several times in order to get that well localized. It may take a long time to probe out several cognitive functions. So if we're in the clinic and we're doing fMRI and we're going to cut into your brain and we don't want to disrupt motor areas, we don't want to disrupt language areas, we're going to run several tasks that have you come up with rhyming words or name objects that you see, and each one of those is going to take 3 or 4 minutes. And then after about an hour, we've really only localized visual areas, motor areas, and a couple of language areas. We've missed a whole bunch of other areas. So it'd be better if we could see lots of different functions all at the same time. And this is what's led to resting state connectivity domains, where we can kind of see which neurons are wired together and firing together, and how they're communicating with each other. And we can see networks in the brain. So things that are performing like functions, even though we haven't given you a task, we can look to see things that are communicating with each other and forming like functions. So, but what we have to do, we have to record for a long time to see those signal variations, and we can then see what's correlating with each other, okay? So instead of showing you flashing checkerboards every once in a while, we're just gonna have you stare at a plus sign for 10 minutes or more. And we're going to tell you, please stare at the sign for 10 minutes and don't do anything at all, okay? Your brain is not going to do nothing. It's going to do a variety of things, but we're going to look to see what's actually communicating with each other to pull out networks from that. But that also includes don't fall asleep too. That's a tough one. So not so much resting state, but not task fixed, okay? We're not giving you a fixed task to localize functions. We're letting you just stare at a plus sign and your mind is going to wander and do what it does. So I put this in here. The resting state folks is a really strong community with very strong opinions. And so I put in a protest here, no forced tasks. So functional MRI without a task, how does it work? Let me show you a little bit how this works. And what we're going to do is we're going to acquire slices, so axial slices of this brain stacked all on top of each other to get a 3D volume. We're going to acquire that every 1 to 2 seconds all the way through. And then we can look at a pixel somewhere in the brain, and we can look across that 10-minute scan. We can look and see what the value was everywhere. We can look at another pixel and look at it, what its value is everywhere. And then we start to see, hey, these actually look like they are communicating with each other. They're highly correlated, so they're probably connected. And we can make a map of what regions in the brain are connected to this. And then we have that as a network, okay? And so this network is groups of pixels that are talking to each other. And we have lots of different networks we can look at. And they are well repeatable across subjects and give us a lot of information about different functional organizations in the brain. Okay, and here's another look at that. This is a dense connectome from human connectome data and showing what's all connected to pixel 1 here, and then what's all connected to pixel 2, which is somewhere in a somatosensory cortex here for pixel 2. Okay, so we can map out the functional regions, how they're connected, and we can map out these networks and understand what's connected to what. So I hope at this point you understand well what is fMRI, how are we measuring function in the brain, and then what is this resting state? What are we measuring? It's still that same function, but it's not with a specific task. We're just looking to see what parts of the brain are correlated with each other. So it would be great if that was, the most of the story, but it turns out that there's a lot of signals in this data that are not related to brain function. As we're collecting these images every 2 seconds, there's other things that are showing up in that data, not just brain function of neurons talking to other neurons somewhere else in the brain. And that can include motion. It can also include physiological noise, okay? So we're looking at a blood-based response. So certainly cardiac pulsations are going to cause some signal variations in that data too. And respiration does too, where we're imaging in a magnetic field, and the movement of your chest wall up and down does affect the signal in your brain too. So these confounds may be actually consistent across groups of subjects. There's been a lot of work showing that some of these things change with age, with certain patient groups, with certain populations, that you may find that the motion or the physiological noise has some correlation across groups of people. And so now suddenly we're getting relationships in our data that are reproducible across subjects that may be different across groups. And that starts to cause concerns that as we look at resting state connectivity data, there may be components that are coming out that are different between groups that are not really related to brain function. So we want to be careful about that. And it turns out what makes it worse is that fMRI is not acquired as the whole brain at once. It's acquired as a set of slices, okay? So we acquire one slice at the beginning. Let's say we're acquiring every 2 seconds. We acquire one slice at 0, and that last slice gets acquired 2 seconds later, okay? And so we're scanning through the brain to get this function, and we're not sampling at the same time. So if you have something like physiological noise or cardiac pulsation, these things can be spaced out in time, and I may be sampling a cardiac pulsation differently at one part of the brain than I did at the other. This is a technique that we use that I'll talk about at the very end of the talk called SimulScan, where I can actually see dynamic activity while I'm doing fMRI scanning. And I just simply rotated my eye, looked up, looked down, looked up, looked down, and you can see the eyeball rotating there. So, but the main reason why I show you this is so you can see when we excite a slice, it takes a while for that signal to recover. So we're actually able to see which functional slice just got acquired in this acquisition, which has a TR of 1.5 seconds. So it takes 1.5 seconds to acquire the whole brain here. All right, so we have some issues, and I want to point out something that I have no evidence to support, but I think is also going to cause this to be reproducible, and that is that modern scanners have this function called auto-align, where if I put you in the scanner today and I put you in the scanner next week and next month and a year from now, it actually finds some certain anatomical landmarks and it prescribes the slices in a reproducible manner. So if I generate information that certain regions of the brain are correlated with each other just because they're sampling this physiological noise at different times, it becomes something that could be very well reproducible in yourself and reproducible across populations, just because of the fact that we're scanning very similar slice locations and they are spread out over time. Okay, so how do we get around this, and what do we need to do? Well, I'm going to show you one more example. There's a lot of literature showing how these physiologically induced fluctuations look a lot like resting state connectivity data. And so this is a paper that's frequently cited that started looking at a lot of these things quite a while back now. But this is looking at one subject and showing correlations with the respiratory volume per time curve and showing how the t-score corresponds with the shift in time of this respiratory volume curve. And so we can see negative and positive correlations showing up in very specific regions in one subject, but then even across a whole group, we get highly reproducible maps here, okay? And so these things are preserved across groups and they can result in signals that can look a lot like connectivity signals. So the way we're going to address this question is we're going to image very, very fast. And I'm going to do it in a way that I can then freeze the physiological motion across the volume. Okay, so we're going to try to image full 3D all at the same time, get a whole picture of the brain. And then we're going to image so fast that we can go back and mimic the way standard fMRI samples data. And then we're going to show how that leads to some additional components detected that are likely due to physiological noise. Okay, so how can we image faster? I'm going to tell you about the technology first, and then we'll show you the application of it. So we have a high need for speed here, but in fMRI, we need the whole brain coverage. We have to cover every slice of the brain, and we need it fast, at least at 1 second per volume And normally we do 2D imaging slice by slice. Some new protocols can do a little faster. We can acquire maybe 2 slices at the same time or 4 slices at the same time spread out. But compared to the cardiac pulse waveform going through the brain, this is actually pretty slow still, okay? So we're using techniques developed by Jipei Liang, who is here, and gave a talk very much about this in the beginning of the week too. So we're using his partial separability model, which is going to unlock for us an order of magnitude increase in speed. And this is really what we need. It's going to improve the quality and the coverage also at the same time. So this is a really great technology. It's not just a framework, but it's also suggests ways to acquire data. And we're leveraging all aspects of that. And we've designed a sequence to really make good use of this model. And we've been using this for dynamic speech imaging. So I'll show you one example of that when we get to that point too. It's enabled us to do things that no one would thought were possible, and our reviewers still give us a hard time about. So sampling this data is really difficult. We want 2 millimeter isotropic resolution. I'm not going to show you that today. I did acquire it, it's too big. I cannot even do it. I'll tell you about that at the end. But we would like to get 2 millimeter isotropic fMRI of the whole brain. That means a matrix size. So how many pixels we'll have is 120 by 120 by 60 pixels going through the brain. We need those sampled very fast. And if we do that, we would have 10 million complex valued samples per second if we wanted to try to do this. And it turns out our scanners are not able to do this. There are safety limits on the gradient performance. And even if we didn't have those, we still wouldn't be able to go fast enough, because we have to, in order to get this functional signal, we have to wait a certain amount of time in order to build up that contrast too. So we need a different way of sampling. We can't just go through and sample all these points that we need to make these images in a series. We can't get these enough samples. So we have to have some other way to do this. And this is where we're going to use the partial separability model. So it turns out the way the partial separability model works is it looks at pixels in the brain and it sees that there are spatiotemporal correlations existing in our data. Not every pixel is unique and uniform. And so we can leverage this. We don't need to image 120 by 120 by 60 pixels because there isn't that much independent information in the image. And so what Jipei did back in 2007 is develop this model and recognize that most of the biological signals we look at are separable, partially separable. And so he developed this model, and this is the only equation you're going to see over and over again. And so I'll explain it, but he developed this model where we can represent, you can think of K as our imaging location here. That's perfectly fine. And T is time. As we go across this 10 minutes of our time series, we really only have a set of spatial maps and temporal components here. So you can think about this as a couple of different ways, as principal components or independent components of our image. And we can represent our full 4-dimensional, because we have 3-dimensional space and 1-dimensional time, our 4 full-dimensional object as a set of 3-dimensional images that are modulated over time and added together. Okay, that's the sum, they're added together. And we're going to be able to represent most of our signals very accurately with very small numbers of images weighted in. Okay. So what is a separable signal? You could separate out in all 3 dimensions of our image, X, Y, and Z. And then you can have a time function. But what we're going to do is we're going to have this, we're going to have spatial maps. We're going to have L of them. L is going to be a small number. It's going to be the rank of our low rank approximation here. And we're going to have a 3-dimensional image weighted in by time at each location. So we're going to be able to turn in thousands of images, turn them into tens of images that are weighted over vectors over time and represent with very high degree of accuracy our full 4-dimensional data. I want to give you an example of how this works, okay? Because this does seem a little bit strange and it may be a little unusual. And so I'm gonna put it not in MRI terms here, but hopefully in other terms. So this is a movie. This is a clip from a recent popular movie. So it's 1080 by 1920 by 431. So of course that's Dr. Strange. So hopefully you pick up on some of the play on words on the next couple of slides, okay? Otherwise, it's just going to be boring still. So this is a clip, 1080 by 1920 pixels in this image, and there are 431 images that are put in a row, okay? So 431 frames, and it's 60 frames per second for this clip that I pulled down from YouTube. So very complicated image, okay? There's background, really high resolution structures in the background, and they are moving, okay? And he is moving and moving his arms and talking and yelling here. And so if we think about how many points of information we have here, we have 1080 by 1920 by 431 frames. We have 894 million points in this movie. So what I'm going to do is show you that I'm going to pause this first. I'm going to show you the same thing, but I'm going to use a rank of 35, and I'm going to represent the same movie. So instead of needing 894 million points, I'm only going to use 72 million points. And that is that I have 35 frames, and that's it, only 35 frames, not 430 frames. 35 frames of data that I simply weight differently each time point. And you're going to see that despite this being a very complicated scene, this has got to be a worst case scenario. We are capturing a lot of the dynamic motion here. I'm going to play it again in this scene. And so this is really representative of what we're doing in our acquisition then. Okay, so a reduction factor of 12 in the storage here, which is, that's more than order of magnitude. So we get spatial basis functions and we get temporal basis functions, but basically we take each image and we weight it by temporal function to get each frame. There's 431 frames. And so I take my 35 images and I simply weight them by this time function. And when I add them back together, you get the movie that you just saw. Okay. So it's pretty amazing that such a small amount of information can represent that image so well. And this works really great. And Jipei, I'm going to show 2 slides from Jipei here. He's been able to use this to do a number of amazing things. So cardiac imaging, looking at the blood flow and dynamic heart. And this one even has an arrhythmia. It's not a regular heartbeat. And so we're not relying on things being completely reproducible and repeatable and periodic. None of that stuff comes into play. We're able to get the really good representation of the 4-dimensional data. And then the talk that was given on Monday by Jipei showed some of the metabolic information. So there's this other dimension. When you get a structural image, you can get metabolic information out. And we have different metabolites in the brain and they have different frequencies. And so you can read out in that direction, you can get this. These are really high dimensional data that requires lots of scan time. And in a very short time, he's able to get really high resolution and very high quality information about chemicals and how they're distributed. And in a stroke, we can get lactate buildup and we can get loss of NAA, which is a marker of cell membranes. So very exciting information, quantitative information that you can get out of these techniques to be able to understand how this works. Okay, so that's great. If I had my data and I needed to store it in, oops, I get echo here. I'm going to mute somebody if I have the power. There we go. All right, so that's great. If my brain imaging data is taking up too much space on my hard drive, I can compress it. But how does that help me acquire data? Okay, I just told you, like this movie, we can compress it down. But that would mean that we had the movie to begin with, right? So the nice thing about the partial separability model is it actually provides a way for us to sample data in which we don't have to have the full data set to start with. We can actually sample things separately. So this is what helps us go fast. So we separate space and time. So hopefully some people get that reference here too, why we use this particular image. So we break through, the breakthrough from the PS model is that it enables us speed ups by separately sampling temporal and spatial resolution, okay? So we have these 2 terms that are put together here. We have a spatial term and a temporal term. We can sample them separately and put them back together. So we're basically imaging the compressed data directly. And that is what has allowed us to make huge advances, because we don't have to get the 890 million points. We can actually just sample the 72 million from the first part. And the way this works is we interleave a data acquisition scheme. And so we take imaging data with, so first of all, we take navigator data. So we keep repeating some really, it's really a twisted cone in 3D that we sample. We don't have to make images out of this data, so it can be just some arbitrary trajectory. But every time we go to collect data, every single line of data we collect a navigator, and that provides the temporal information. And then in between this, once we know the temporal location we're at, we collect some imaging data, and we can sample that however we want. I show here Cartesian imaging, where we collect just one line of K-space each time. In the fMRI data I'm going to show you, we actually require a spiral, one KZ line of a 3D spiral in our imaging. But we can actually separately design how we're sampling time and how we're sampling space in these acquisitions, and we can make it work really well. We actually acquire both of these at the same time inside the same TR, and that helps us to go much faster. And so, yeah, I'm not sure this, let me just get all the pieces to show up since. Okay, so what we do then is we take all the temporal navigator data and we put it into a matrix, and we pull out the temporal basis functions from this by looking at some of the components of the singular vectors in this data. and we can pull out the temporal components, the top L of those, if we chose a rank of, like, for example, in that clip from Spider-Man, we chose 35. And so we pull out the 35 temporal basis from this, and then using those temporal bases, we put them back into an estimation program where we can then estimate the spatial maps that go with those temporal bases. And so from this, then we pull out the 35 spatial maps that are represented in that data. And then to get back to the original data, we put these all together. We take our temporal bases and our spatial bases, we mix them back together, and we get our full movie back. Okay, and this is showing you an example from speech. So we can apply other constraints on that data too. So we can apply extra constraints to other components here, and that may help us out for different purposes. So, and we put that back together. I'm going to show you one example from speech, and we put that back together. We can do things that are not possible. And I like showing this example because when I started doing speech, we were able to do one partial slice, only 1/4 of the field of view here. And we were able to do 20 frames per second of just a single 2D slice of speech. And now we're able to do a whole 3D volume. This is an isotropic 3D volume, and at 40 frames per second. And this person is going to say the rainbow passage. And I don't think I shared the audio on the Zoom call, so you probably can't hear, but we're able to see in all three directions. We're able to make measurements of how things are moving and be able to see the full three-dimensional structure, dynamic structure in all these directions. So we're covering 10 times as many slices as we started with. We're covering it 10, we're able to get up to 10 times faster. This is 40 frames per second, but we're able to go much faster than that. And the quality of the images has actually gone up quite a lot too. So these are almost order of magnitude in 3 domains improvement over the way we were doing things before. So, but we want to go fast in fMRI. And so I'm going to show you that here. So we're going to show you the application to looking at, if you remember, we were saying we need to be able to image fast to understand the physiological noise in these functional signals. The brain signals don't go faster, but the way we're sampling the data is actually too slow right now. So we're going to go back and we're going to fix this issue that in standard fMRI, we're acquiring 1 slice at a time. It takes about 1.5 seconds to get all the way through. And timing differences between the slices may cause some signals and correlations in the data to occur. So what we're going to do is we're going to actually acquire the whole 3D volume very fast. We're going to acquire 60 millisecond TR, and then we're going to be able to go back and simulate what a normal fMRI acquisition might look like from these. Okay, so this is our early results. We got this working. We ran this one here. I'm going to show you. I have a much higher resolution data set of 2 millimeter isotropic. And we just ordered a computer with more RAM because we're not able to actually, we're able to reconstruct it, but we're not actually able to load the results and analyze them. There's way too much data. So, but this is 3.8 by 3.8 by 3 millimeter resolution. We have 40 slices going across the brain here. And our TE is 25 milliseconds, which is very similar and typical for a fMRI acquisition. And our TR is 60 milliseconds. And in that 60 milliseconds, we get all 40 slices. Okay, so we're covering all the brain at once. We say slices here, but it's a 3D scan. It's not a 2D scan. And in a 5-minute scan, I get 5,280 time points. In a typical 5-minute scan for fMRI, I get 100 or a couple hundred. So this is definitely a lot of data to work with. And then we're going to do a visual task for this one, 20 seconds on, 20 seconds off, 8 repeats for the visual task as we go through. And so just want to give you a look at what the data might look like. So this is some correlation with the task, and I show a single pixel here as waveform. We get this really high resolution, high temporal resolution signal waveform associated with that too. And this is sampled so fast that we can take a look at the spectrum. So we've sampled the data with sufficient speed that we get up to 8 hertz of information here out of the data. And so we can take a look and see how certain physiological signals or task signals show up. Most of the time, our task signals, due to the fact that our hemodynamic response, our blood oxygenation response is so slow, those task signals usually show up at about 0.05 hertz or so, up to like 0.15. But what we have here is we have cardiac pulsations, we have respiration, and we have that showing up in our data. And you can see that here. So this person had a heart rate such that it's about 1.3 hertz, and we can see 1.3, 2.6, and we can see up to about 5 harmonics in this data. I zoom it in here, and you can actually see the signal here. So we're able to actually see up to about 5 harmonics in the data. And there's a lot of other information here too. So we'll go through and see that. So, but our main point was to try to look and see how this sampling impacts the physiological noise and how it shows up in the data set. And so I want to show you what we did here. And like I said, this is new and we have this speed and we were like, okay, what should we do? And so if people have better ideas, please feel free to contact me afterwards and let me know what you are thinking. But I want to just point out what we did. So this is the 40 slices that we might do in a typical fMRI experiment. We might acquire these separately. In our experiment, we're going to get them all together, but I color coded them here. And so in a standard fMRI, it would take us 40 separate acquisitions to get that full volume sampled, okay? And we would get just one image, okay? Just one image in that same time. And then we would go to the next time point and we have to acquire those 40 slices again. And we would get a second image. And then we have to acquire those 40 slices again and we get our third image, okay? In ours, we get for every single one of these little bins here, we get all the slices sampled. Okay, so I get all of them every time. Okay, so I'm going to end up with 40 times more volumes than we would get in the standard. But what I can do from my data is I can simulate a couple of things. So I can simulate a standard acquisition. What I can do is I can take this slice from this volume, this slice from this volume, this slice from this volume, and so on. And I can build up what the standard fMRI would have looked like for my acquisition, where I've frozen the information across all the slices, okay? So that's standard one. I can also do something just to make sure that there's not some really weird representation in that data. I can shift over. I shifted over. I sampled the last slice first, okay? So purple slice first, then blue, then orange, then red, okay? So I did another acquisition where I shift to a different slice. Okay, then I can also do the ideal situation. The best possible thing you can do, I can sample all slices in the same volume, in the same one of these subsets, and I can wait till the next image would have come out, and I can sample all those slices in that same subvolume again. These are all at the exact same time sampled. So if there's any physiological noise, it's all sampled at the same time inside one of these volumes, okay? So I can do that, and I can choose which one of these bins I want to actually produce that image in. So here's synchronized sampling 2. I chose something out here, okay? Halfway across this, okay? And I stack all those slices together. And then finally, I can do one more thing. I can take my data from all of these acquisitions and I can average over the whole window. I can just add everything up across the whole window and make one image that way, where if there is physiological noise and it's varying across these, I've kind of collapsed it across so that every single slice has the same contributions of that. They've sampled the entire period across, or the entire sample across that time frame, okay? And so I can put that together. I'm going to call that average, okay? And average is probably the actual best case scenario because it's not just fixed on one particular physiological noise. It's actually averaging across all the different representations of the physiological noise. And the TR I'm going to end up with here, because I have 60 milliseconds for each one of these, if I have 40 slices, the TR I'm going to end up with is 2.4 seconds for the simulation. And so across 2.4 seconds, I have a full heartbeat. And so I'm going to be averaging across the whole cycle and then some, okay? So hopefully this makes sense. It's a very complicated slide, but it's necessary to understand the next page. So what do we expect? Let's talk about that. So my hypothesis to you at the beginning was that because of the way standard fMRI is sampled, I am getting correlations in my data because I'm sampling physiological noise at different times for different slices. And so I would expect to get extra components coming from this just because I'm sampling the physiological noise differently. So if we look at our acquisitions, we would expect a higher number of components. We're going to look at networks, and we're just simply going to count the number of networks we find using a technique called using an ICA technique called Melodica in FSL. We're going to count the number of components it finds. And so we would expect to find a higher number of components for standard 1 and standard 2. We'd expect to find the least number of components for average here, because we're averaging across all the physiological noise across more than one cycle of it. And so we expect this to be, but I do fully expect that synchronized 1 and synchronized 2, we've frozen those, and I expect those to look very similar to average here, okay? So that was our hypothesis, and it turns out that's what we found. So we have here the number of components we found across these different components. So we have average here, we have 34 networks that were found in the data. And when we look at synchronized 1, 2, and 3, we find very similar numbers. They're just slightly higher than average, but somewhere really close to the 34. And then when we go to the standard sampling, we get much higher number of components. And it makes sense because we've incorporated, this sampling where it's different on different slices, and that's adding a lot of additional components into the data that are not in these other techniques. Okay, so if we look at some of those components, I can look and see what comes out of some of these components from the standard sampling. And what we'll see is some correlations that are, largely focused on individual slices, probably related to physiological motion here at the back of the brain, or motion. And then there's some correlations in a slice somewhere else too. Okay, so it's got some spatial structure to it that's based on sampling. And we see a blue one here, which is component 27, which also has some spatial structure to it too, where that was picked up as some physiological noise sampled in that slice that correlated inside that slice more than it does with other slices. Okay, so hopefully that makes sense. I'm going to tell you where we're headed with this. I would like to talk to people who are interested in the high-speed fMRI and have ideas about how else we should be able to use it, realizing that brain functional signals are not there. We've tried, we've looked just to see whether we have enough sensitivity to see things that are happening quickly. And of course, that hemodynamic response, the bold response function, is way too slow. There's not additional information there. But we would like to talk to people if they have more ideas. What we think we're going to do next is look at some of the physiological noise correction schemes, and we should be able to provide a best case scenario of looking to see how effective those different schemes are. We're also going to use the technology to do an additional task. So I had showed you SimulScan at the beginning, where we scan these dynamic images at the same time we scan functional brain scans. And the dynamic images we used back in 2011 when we originally did this were sufficient to see tongue motion, but insufficient to actually measure aspects of the motion. So this is our picture from our paper back in 2011. We told the subject to rest and then tap their tongue, and rest and tap their tongue. And if we look at a strip plot, just take a strip of pixels through the tip of the tongue, they rest and then they tap, they rest and they tap, they rest and they tap. There's enough information here to see that something is happening. And then we took brain slices, and we can see the brain function associated with those. And we were really surprised when we did this the first time that we saw a little blip here, and we saw brain activation going with it. And we went back to look at the movie, and we saw the subject swallowed. And so we picked up an incidental swallow and the brain activity associated with it. And so this goes back to that resting state is not task-based. We didn't give this person a task to swallow, but eventually we redid this experiment where we told people just to sit in the scanner and watch a movie. And we didn't tell them what we were doing, but we were actually watching them swallow. Okay, so, and then we pulled out all the brain activations associated with natural swallows. So we're hoping to do this in stroke patients. We just submitted an R1, and we're going to merge together our high resolution, our high temporal, spatial temporal resolution dynamic imaging with our high spatial temporal resolution fMRI. And we're going to be able to pull together this one platform to be able to study swallowing in stroke patients looking for brain functional deficits and motor deficits in the movement. This is really critical for neuroscience because we're going to be able to evaluate how different parts of the brain are involved in controlling the motion, sensory motor integration aspects of it too, and be able to see exactly what that leads to, what deficits in the brain lead to deficits in the actual motion related to swallowing. And it's really important to be able to do this activity with natural swallows because stroke patients, it's not safe oftentimes to give them something to actually swallow. And so we're able to just use incidental swallows, which are still happening in stroke patients, and they happen naturally. And so we're able to use those directly. So with that, I'm going to stop here and take some questions. So hopefully you will understand that I gave you a talk that's like maybe some early stuff here that hasn't been all the way thought through. Wanted to do that to give you an opportunity to see what happens and how these things work and how we develop new technologies. But very excited to take any questions you might have. This was a big project involving lots of people from lots of places. And we have some grant funding to do dynamic speech imaging. And hopefully we soon have grant funding to do speech or swallowing and fMRI at the same time. So thanks very much.